Hello, biology students! Last time we were building the start of our DNA foldable and learning about DNA structure. We built this foldable using this worksheet, and we filled in polymer A and polymer D by folding it up, and we looked closely at the fact that a nucleotide, the monomer of DNA, is a sugar, a phosphate, and a base. Phosphates and sugars are on the outside, bases are on the inside, and we used our base pairing rules to know what should go on polymer D. This second half, we're going to be looking at the next page in our packet, which is how do we then replicate this whole double helix. This is a double helix if we were to twist it up, but we've untwisted the DNA double helix so we can see it flat like a ladder, but it has two strands. That's the double part. Helix means twisted. So how do we go from one double helix to two double helices? That's the process of DNA replication. They need to be exactly the same. So let's read our directions. In this first direction, it says that the actual enzyme that speeds up chemical reactions here is going to be the enzyme helicase. Notice that helicase sounds like the word helix, like DNA double helix. It is in charge of unwinding and unzipping the helix. So pretend it was already all twisted, it untwists it, and what would unzipping look like? Well, think of this as the teeth of a zipper. Unzipping would actually look like pulling it apart. And when we pull it apart, we can think of these two original strands, A and D, as now templates, just like how A was a template originally for making D. So the next piece of direction says, then act like the DNA polymerase, the enzyme, because it's ACE at the end, that makes more DNA polymers. And you are going to use your complementary base pairing rules to make complementary strands, remembering that A always goes with T, C always goes with G. Apples in the tree, car in the garage. Okay? And so we are going to use the original strands as a template for making new ones. So we're going to label B and C as new strands. New strand, new strand. So let's focus first on B here. So that I don't get confused, I'm going to fold it in half like this. And let's build B first. We're going to use A again as a template, remembering that each of these is a nucleotide. So we have a sugar and a phosphate and a base.